Hello everyone. My name is Suman Verma. I'm your biology faculty, and I'll be teaching you botany. So today's topic is principle of inheritance and variation. So in this, we are going to learn about how genetics are involved in the inheritance and how we are getting variations from our parents. How we are different from our parents. Okay, so let's start. So our key points, what we are going to study in this chapter are Mendele's law of inheritance, inheritance of one gene, inheritance of two genes, sex determination, mutations, and disorders. Okay, now first, what do you mean by genetics? So genetics is nothing but study of inheritance and variation. Now, what do you mean by inheritance? So whatever the character or whatever the genes you are inheriting from your parent, that is called as inheritance. That means I can say from parents whatever we are getting to progeny is considered as inheritance. Now what is variation? Variation is nothing but degree of differences. Means we know, obviously, we are not that much we are similar to our parents, but we are not exact copy of our parents. So that difference is called as variation. I hope everyone are clear with inheritance and variation. Now next, so for this for this, okay, so for this we Mendel, he was a scientist who proposed some law of inheritance. Okay, so his full name was Gregor Mendel, and he conducted hybridization experiments on garden peas for 7 years, from 1856 to 1863. Proposed the law of inheritance in living organisms. So Mendel is the scientist who proposed the law of inheritance. Now, in the upcoming slides, we are going to learn what is law of inheritance as well. Okay, so what he used for his experiment? What he used for his experiment? First thing, he used static analysis and mathematical logics. Yes, he included maths and even statistics to prove his experiment. He used two opposite traits. Opposite traits in the sense, if, for example, character, he is using about shape. Okay, but in shape, he used two traits. That means one will be wrinkle and one will be round in shape. Okay, so two traits, opposite traits he used. Next, large sampling size. Obviously, he used very large sample size. Okay, now one character with contrasting trait. Yes, one will be dominating trait and one will be recessive trait. Okay, next. 4 true breeding pea plants. 14 true breeding pea plants he used. Okay, now next. So let us talk about inheritance of one gene. Let us talk about inheritance of one gene. So in this, what happens? We already know. We know that genes are always in pairs. Yes or no? Genes are always in pairs. So you can see here gametes. So this is parent 1 and he is tall, and this is parent 2, that is tall. And always remember, whenever we are taking any parents, the parent should be in homozygous condition. Homozygous condition. Why homozygous condition? Because we need gametes. Okay. Now, so from tall parents we got gametes which are capital T and capital T, and from dwarf parent we got small t and small t. Now we are going to cross them. Okay. Now when we are crossing them, obviously from this t we got one t, and from this t we got small t. So capital T and small t. So all the progeny, whatever we are going to get here, are going to be capital T and small t. That means all the plants are going to be tall. Why tall? Because you can see this tall parent. Tall parent here is the dominating parent. Here is the dominating character, or we can also say dominating gene. Okay, so this will come under F1 generation. So I can say under F1 generation, all the plants were, all the plants was tall. Okay, now what we will do from F1, from F1 we will take the parent. Now we will do selfing. Selfing means for this F1 generation, we are going to cross it with same F1 generation. Means both the parents will have one capital T and one small t in their gene. So what are the two gametes we are going to get? One capital T, one small t, one capital T and one small t. Now we are going to cross them. Okay, by selfing. So you can see this square. This is also called as Punnett square. So here when we are writing capital T, small t, capital T, small t. So here, when this capital T will be crossed with this capital T, we are going to get capital T, capital T. Here, capital T and small t are going to cross. Here, small t and capital T are going to cross. Here, small t and small t. So here I can say, in genotypically, I got capital T only one. I got capital T two. Sorry, not this. Okay, so I can say genotypically, I can say I got capital T 1, capital T small t 2, and small t 1. So my ratio was 1 ratio 2 ratio 1. Okay, now phenotypically, mine means with the appearance, if I say, obviously capital T will be tall, capital T small t will be tall, capital T small t will be tall, and small t will be dwarf. So my ratio will be 3 ratio 1. So this is my phenotypic ratio, and this is my genotypic ratio for monohybrid cross. Means for monohybrid cross. Monohybrid cross. Monohybrid cross means here I am only going to use one character and I am going to do this. And always remember, whenever we are doing any hybridization, always take parent. Parent should be in homozygous condition. It should not be in heterozygous condition. Okay, now yes, next. Yeah, so in monohybrid cross, we are going to learn about test cross. So what is this test cross? So Mendel crossed the tall plants of from F2 generation. In F2 generation, whatever the tall plant are there, it might be capital T small t or it can be capital T capital T. These are crossed with recessive character. These are crossed with dwarf plant. That means recessive parent. Okay, this is called test cross. In a typical test cross, an organism, or in this case we can say pea plant, shows a dominating phenotype and whose genotype is to be determined. Is crossed with a recessive parent. Always it needs to cross with recessive parent. That means small t, instead of self crossing. Okay, so here we took tall parent, tall from F2 generation. This is from F2 generation, and in that with that we are going to cross it with small t and small t. Okay, so when we are doing it, we got our cross that 50% will be dwarf and 50% will be tall. So this is our phenotypic ratio. Okay, now so this is for the test cross. Next, laws by Mendel. Yeah, so just now we have learned that Mendel proposed a law. That law is nothing but law of inheritance. So under law of inheritance, we have two laws. My first law is law of dominance, and my second law is law of segregation. Okay, law of segregation. Now we are going to learn about these two laws. These two laws. Next. So first let us study about law of dominance. Let us study about law of dominance. So in this what happens? First parameter of this law is that characters are controlled by discrete units called factors. Here factors are nothing but genes or alleles, we can say. We can also say alleles. Okay, gene and allele and factors, these are same. So obviously all the characters are controlled by who? It will be controlled by genes. Yes, genes occur in pair. Obviously we know that all the genes are occurred in pair. Just now we have studied, if that there is a tall plant, it is denoted with capital T and capital T, means it's in a pair. Okay, now in a dissimilar pair of factor, one member of the pair dominates the other, means recessive, obviously. So here what happens, if there are dissimilar pair of factor, means it is capital T and this is small t and small t. Dissimilar pairs are there. Obviously, one is going to dominate the recessive pair. Okay, so this is the meaning of third law. Now, as you can see here, capital T and small t are crossed. Then what happens? We got all in F1 generation, all the plants were tall. That means what happened here? The capital T dominated the small t character. Okay, so and again we took a heterozygous tall, that is capital T and small t, and we did what? We self crossed it, right? Self crossing we did. Then with that, what happened? We got F2 generation where three plants were tall and one plant was dwarf, which was a homozygous condition. Next, this is about the law of dominance. Next, now, uh, let let us study about Mendel's law of segregation. So, in law of segregation, what happens? Uh, Gametes receives one alleles from each. So here what happened? You see, parents. One parent is in YY condition and one parent is in small y condition. That means they are telling each homozygous parent in the parental generation forms only one kind of gametes. Yes. If it is a small y, obviously the gamete will be small y only. It is not going to give me any capital Y. Okay. Now, so the heterozygous F1 offspring forms two kinds of gametes. Now what happened? Obviously, one gamete from one parent and one gamete from my other parent. Okay. So in my F1 generation, what condition I am going to get? I am going to get heterozygous condition. Means here I am going to get two kinds of gene. That is a capital Y and a small y gene. Okay. Now, when I did selfing of what? Selfing of F1 generation. Means capital Y, and small y. Here what will happen? Self-pollination of the F1 offsprings forms F2 offsprings with a 3:1 phenotypic ratio of light violet to light orange seeds. So here, what happened? Phenotypically, we got what? Phenotypically, we got 3:1. That means 3 became tall and 1 became dwarf. So this is the study we are going to do in law of segregation. That means both the parents are involved 50-50%. Okay, both the parents are involved 50-50%. Now, yeah, next comes law of dominance. Next comes law of dominance. In law of dominance, incomplete dominance. Sorry, we can yeah, law of incomplete dominance. So in incomplete dominance, what we are studying is when experiments on pea plants were repeated using other traits in other plant, it was found that sometimes the F1 had a phenotypic that did not resemble either of the two parent and was in between two of them. Means here I can say that in incomplete dominance, neither of the parent become dominants. That means, see, here you can see I have capital R, capital R, capital R and capital R are denoted for what? Red color. Red color, red flower. Okay, and smaller resembles white flower. Okay, now with this what happens? Because of this, what happened? Now, when we crossed, obviously one gene means one capital R will be coming from the one parent and one small r will be coming from the other parent, right? So now, actually, as per the Mendel rule, what we have to, what should happen here? This flower should be in red color because one trait will become dominant. Yes or no? But here what happened? Neither of the parents were completely dominating the offspring. Okay, it was in between. It is in pink color. Okay, it is in pink color. That means it's in between. That means it's an incomplete dominance. Incomplete dominance. Okay, because it was in between of the two. Neither red color got dominated, neither white color got dominated, but it was in between. Okay, the F1 generation was in between. So this is called as incomplete dominance. Okay, now next, um, next our next our next topic is codominance. Our next topic is codominance. In codominance, what happens? In codominance, both the parents are expressed. Okay, both the parents are expressed. The best example for codominance is blood group. Is blood group. It's the blood group. So in blood group, what happens? Obviously, we only have three types of blood types, right? A, B, and O. As you can see in the screen, we have only three types. That is A, O, and A, B, and O. Now, in type A, we have antigen B, sorry, anti B, antibody B, and we have antigen A. Okay, we have antigen A and we have antibody B. Okay, now in type B, if your blood group is B positive or your blood group is B, okay, then what happens? You will have antibody of A and antigen of B. Okay, now if your blood group is AB, then what happens? You will have antibody of A and antibody of B, and also you have antigen of A and antigen of B. Okay, and if your type is O, if your blood type is O, then you don't have any antibody. Okay, you will not have any antibody. Clear? Now, this is a Punnett square. If you this is a Punnett square for blood grouping. Okay, so with this you can understand how it is dominating, how it is co-dominating. Means both the alleles or both the genes are getting expressed. Okay, here is the Punnett square. Now, so this is about the codominance. Next is inheritance of two genes. Means we also call it as dihybrid cross. We also call it as dihybrid cross. We call it as dihybrid cross. Now, so in dihybrid cross, what happens? In dihybrid cross, we use two different characters. Okay, for example, here what we are using round and yellow. That means round and yellow both are dominating. It is denoted by capital R, capital R, capital Y, capital Y. Okay, and we are using wrinkled and green. That means smaller, small y, small y. Two characters. So obviously from this, what are the genes I am going to get? I am going to get R and Y gene. And from here, what genes I am going to get? Smaller and small y. Now when I did it when I crossed it, I got capital R, smaller, capital Y and small y gene. This will be round and yellow. Why round and yellow? Obviously because capital R and capital Y are dominating in nature. Now, as we know, if we got F1 generation, what we have to do? F1 generation should undergo selfing. Should undergo selfing. Okay. Now, when we are selfing, this is the ratio we are going to get. This is this is a Punnett square which we are going to get. Okay, so phenotypically, what is the ratio? Phenotypically, the ratio is 9 ratio, 3 ratio, 3 ratio, 1. Okay, so in this I can say 9 are round and yellow, round and yellow. 3 are round and green, and 3 are wrinkled and yellow, and one is wrinkled and green. Okay, and this is round and yellow. Okay, 9 are round and yellow. This is phenotypically. This is phenotypic ratio. Now, if you how to know genotypic ratio. Okay, so here I'll give you one short trick to remember that. Here I'll give you a short trick. So yes, yes. So if let us this is phenotypic, right? Let us talk about genotypic. So here is a trick I'm going to give. See, we already told that this is a dihybrid cross. That means two genes are taken and two characters are taken with four different traits. Yes or no? Now, so for one cross means for monohybrid cross, what is the genotypic ratio? The genotypic ratio is 3 ratio. What is the genotypic ratio? 3 ratio. Sorry, not this. Sorry, one 1:2:1. Yes or no? So this is the genotypic ratio, because in monohybrid cross this will be the genotypic ratio. Now this is for one monohybrid cross and other one 1:2:1. This is another monohybrid cross. Now when you cross this, you will whatever the answer you are going to get, that is your genotypic ratio for dihybrid cross. That means directly you can multiply. You can just multiply and cross check your answer. You can directly cross check your answer. Here you can do like this. 1 into 1, 1. 1 into 2, it is 2. 1 into 1, it is 1. Next 2 into 1, it is 2. 2 into 2, it is 4. Next 2 into 1, it is 2. Next 1 into 1, it is 1. 1 into 2, it is 2. 1 into 1, it is 1. So this is the ratio for a dihybrid cross. For This is the ratio for genotypic which shows dihybrid cross. So this is just a shortcut. In the same way, you can also do it for phenotypic ratio as well. Okay. Now, our next topic is law of independent assortment. So, what is law of independent assortment? So, in law of independent assortment, what happens? The inheritance pattern of one trait will be not affected the inheritance of the inheritance pattern of the another. Okay, now independent assortment occurs during meiosis. Yes. For a single human gamete, the possible ways chromosomes may assort is assorting. Means independent assortment is nothing but what the dihybrid cross. Because in dihybrid cross we have seen all the no trade. One trade will not affect the inheritance pattern of the other trade. Okay. Now one pair of the character is independent to the other. They are not going to interfere. Okay. So that is called as law of independent assortment. No character are getting affected with the other character. Okay. Now see, if you will say, ma'am, but it is dominating. It is dominating, but it is not affecting its inheritance, right? It is not affecting its gene, right? Gene is already present, but it is just dominating it. But gene is not completely eradicated, right? because we can see the same gene in F2 generation. That means the gene is present. The recessive gene is already present in F1 generation, but it is getting dominated because of dominating character. But in F2 generation, what happens? Even the recessive character is getting appeared. Okay, because it is independent. It is not getting affected by the any dominating character. Okay, now next. Yeah. Our next topic is linkage and recombination. Our next topic is linkage and recombination. We already know what do you mean by linkage. Linkage is nothing but physical association of gene. Okay, linkage is nothing but what do you mean by linkage here? Morgan attribute this due to the physical association. Physical association of gene is nothing but linkage. Okay, its term was linkage, describing this physical association of gene chromosome, and the term recombinant to the described generation of non-parental recombination. See, if the same parent are getting combined physically, then that is called as linkage. But if I am getting a gene which is non-parental, means it's not from my parents. Okay, it is any gene which is inserted from outside. Okay, so that will be called, that we can call it as recombinant. Okay, now, so Morgan and his group, they told that, see, if the if we want tight linkage, okay, if you want tight linkage, the recombination will be low. Okay, excuse me. Yeah. So I can say, you can see in the diagram, in the diagram, for cross A and for cross B. Here you can see Y and W, they are linked, right? So they are close to each other. Yes or no? They are closely, tightly linked. Okay. Whereas here W and M, they are not closely linked. They are a little away. They are loosely linked. Okay. So because of that, when I did recombination, what happened? The in cross B, as the distance of the linkage is high, it is showing maximum chances of recombination. But if the linkage is short and it is tightly packed, I can see very less chances of recombination. So this is a theory which was given by Morgan and the groups. Okay, now, so I can say that if linkage is very tight, obviously recombination will be very low. And if the linkage is light, I mean linkage is very loose, okay, or else it might be in distance, then I can say recombination chances are very high. Okay, that's it. This is about the linkage and recombination. Now, yeah. Our next topic is poly polygenic inheritance. So what do you mean by polygenic inheritance? In polygenic inheritance, in polygenic inheritance, okay. In polygenic inheritance, what happens? Many, many gene, many gene will be controlling one character. Okay, many gene will be controlling many character. One character, not many character. Many gene will be controlling one character, and this and for this we can take the example of human skin color. Human skin color. Okay, human skin color, and even human height. Human height. Okay, these are the example for polygenic inheritance where many genes will be controlling one character. Will be controlling one character. Okay, now, for example, as I gave you the example, human skin color and human height. Okay, now, next, next, let us talk about Pleiotropy. Okay, let us talk about pleiotropy. So in pleiotropy, what happens? Single gene, single gene will be controlling many characters. Just this is just opposite. This is just opposite to the polygenic inheritance. Okay, this is just opposite to the polygenic inheritance. Okay, for this, the best example is a disease called as phenylketonuria. Phenylketonuria is a disease which is an example for pleiotropy. Okay, this is example for pleiotropy. Now, because of this is what are the symptoms? The symptoms are mental regard. They'll be facing mental issues. They'll be using hair reduction. They'll be they'll be facing hair reduction. They'll be facing skin pigmentation reduction. Means pigmentation of skin will also get affected, and this is caused because of this one thing which is showing these many symptoms are caused because of one single mutation, one one gene mutation, one gene mutation. Okay, one gene mutation. This is caused because of mutation. Now, okay. Now, our next topic is sex determination. Our next topic is sex determination. Our next topic is sex determination. So already we know what is sex determination. Yes or no? So this sex determination was first given by who discovered this sex determination? Henkin. 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 Okay. In the year 1891. In which year? 1891. Okay. So we already know in human for females it is X and X. For males it is X and Y. Okay, X and Y. So I can say that in male 50% of the sperm carries X chromosomes and 50% of the sperm will carry Y chromosomes. Okay, now next. So female will produce X and X, and if male will produce X and this will go and bind with this chromosome, the progeny will be a female. And if the male will produce Y chromosome and it go and binds with X chromosome, the progeny will be male. Okay, will be male. Now, so in our NCRT, they have given about three organisms, about human, about flies, and about chicken. So chicken in hens this is little bit different, because see here, in humans females are in homozygous condition but males are in heterozygous condition. But in chicken or hen, the females are in heterozygous condition. That means they are in X and Y condition, and the males are in homozygous condition. They are homozygous male. Okay, they are homozygous male in birds. Okay, now, so this is about the sex determination in human. Now, in our NCRT, they also gave us about the sex determination of honeybee. Sex determination of honeybee. Yes, sex determination of honeybee. 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 In honeybee, what happens? In honeybee, the female or the queen, I can say, queen and the male. Okay, so the queen honeybees chromosome number 8. Queen is always in diploid condition, and the male honeybee is in haploid condition. In the haploid condition, that means female chromosome is 32, whereas male chromosomes are only 16. Now, here what happens? Here what happens? Male will undergo mitosis, means equational division, whereas female will undergo meiosis, reductional division. to form 16 and 16 chromosomes. Okay. Now in this what happens? In this what happens? We here if the queen, if the queen, if the queen is giving progeny without fertilization, means without the involving of male partners, then the progeny will be a male only. Okay, because queen have undergone meiosis, so there are 16 and 16. So because of 16, what happens? We are getting a haploid, and haploid is a male. Now, if a queen is involving the male, then obviously 16 from female queen and 16 from male, both will become what? 32. And this 32 is for what? Female. Female. So okay, I can say that if without fertilization we are getting progeny, it will be a male, and with fertilization, if we are getting, the progeny will be a female. Okay, I hope everyone are clear till here. Everyone are clear till here. No doubts? Yes. Now let us talk about mutation. Our next topic is, let us talk about mutation. So what is a mutation? Mutation is obviously sudden change in a gene. Okay, sudden change in a gene. Okay, so now when any DNA sequence alters, DNA sequence, obviously in our gene what happen, what is present in our gene? We have what? We have genes. We have DNA sequence, and in that sequence, if there are any changes or any alteration, okay, that will cause what? Mutation. Okay, it will cause mutation. Now, so this, because of this mutation, it can affect genotypic also. It can affect us phenotypically also. Means by physical, with the physical appearance also, it can affect. With the genetic disorders also, it can affect. Okay. Now, how this DNA sequence can get changed? So this can because of some loss, reduction, deletion of any sequence, or addition of any sequence. Because of addition of any, because there can be addition of any sequence. Okay, because of addition of any sequence, or it can also be caused because of duplication also. Duplication, multiplication, by multiplying, it can duplicate, random duplication. Because of that also, it can cause mutation. Okay, so this is about the mutation. Now, so this is about the chapter. This is about the chapter. I hope everyone are clear with whatever we thought for today's class. I'll be seeing you in the next class with